I'm born on the East Coast. I have always had a passion for the West Coast, which is why I've moved out here now.
I would say first that I work at UCLA, that I'm an assistant professor there. I would probably mention that I come from France as well.
I would describe myself first as a scientist and someone who loves the unexpected and loves doing something a little different every day.
Just say I'm a researcher from North Carolina.
I focus mainly on supporting process chemistry or CMC toxicology, impurity specifications, setting, assessment of genotoxic impurities, and the like. In my former life, I was more of a project toxicologist doing drug development regulatory tox.
Outgoing, definitely an extrovert. I like to be around people. I'm pretty entertaining.
I'm a consultant in the toxicology industry, and so basically I consult doing small molecule drug development, everything from early discovery all the way through NDA.
First and foremost, a curious scientist who just happened to find his way into the discipline of toxicology.
I'm a scientist in tissue applications at Organovo, and I work on liver model development.
So I would describe myself as being passionate about innovation. I mean, that's really, I think that's what the future is.
A lot of things make me smile.
I would say I am a general toxicologist. I am a consultant.
General toxicologist, a non-clinical safety rep on a drug development team with some study directing to fill the cracks in time, assuming they ever exist.
And I'd say for a scientist, I'm on the outgoing side, so.
Very energetic. I like to be around people, so I like the connection and the contact between my clients and myself. I find that really interesting. Been doing this for quite a while, so it's a lot of fun.
I'm self-deprecating. I'm willing to make fun of myself and have people make fun of me and enjoy it.
What I like to imagine myself doing most of the time is brewing beer, but that probably has been turned into a high-efficiency task that I might get to do once a week.
Because I start the day with running, you know, so wherever I am. So you can, if you need some running routes, I can tell you running routes in Tokyo, San Diego, Zurich, but also Guangzhou, Hong Kong, wherever you like, you know, I can take you running.
There is not a lot of outside of work. When I leave work, I usually work from home.
We spend a lot of time in Southern California. It's a wonderful opportunity to get out, to hike, to just explore the beautiful area we live in where it's almost always sunny.
Otherwise, I try to be sane by getting out into the world and do a little bit of hiking as well, just to get some perspective on things, which I find very useful sometimes.
I have a young child, so we also go to pretty much every museum, the zoo. We've been to several zoos actually. Who knew there were so many in the area?
Surfing, golfing, skiing are probably the big three. But, anything outdoors.
Why did I become a toxicologist? Real answer? I finally listened to my parents, and I wanted to have a career that I knew I would be able to get employment, that I would have multiple choices of what I wanted to do, so I wouldn't be, if you can't get a job here, you're not going to be able to work. So toxicology offered me the opportunity to have a job either in academia if I wanted to pursue basic research, which I really did enjoy when I was in graduate school, or government or industry. So I had as many options as I possibly could get.
I actually decided to be a toxicologist in undergrad. I was a student at the University of Oklahoma, and I had a really great analytical chemistry professor named Leroy Blank. I was a pre-pharmacy major, and I talked to him about what I could do to get into the world of drug research.
That's a very distinctive memory. I was probably a 4th or 5th year graduate student. And at the time, in the 90s, there was a lot of us students were concerned about future employment because it looked like the scientific field was getting extremely competitive, especially with NIH funding being cut back, et cetera. And so my graduate advisor came to me with an article about toxicology.
I was really interested in discovering how drugs were made and the process of getting a drug to market and things like that. And he looked at me and he basically said, "You're in the wrong major." So
it was an article in, I think, Scientific American, and it basically said that this was going to be a growth area for the next 20 years, and they were looking for scientists. And so I made the decision to pursue a postdoc in toxicology at the University of Kansas.
So I may disappoint you here, but because my goal was not to go into the pharmaceutical industry, my goal was to, I'm very interested in science, you know, that is something, and then I'm passionate about innovation.
I just joined up with pharmaceutical scientists and started being asked to apply my trade, so to speak, developing these in vitro tools, to which I've been doing ever since. Just so happened to be in the field, in the pharmaceutical field and the toxicological field.
It is something that obviously has a huge benefit for human beings.
I don't think there is a normal day in the life, because I feel this is a good thing, and I actually enjoy my life because it takes me to so many places.
Day in the life, well, you know, you get up and typically go for a run. Like I said, there's not too many bad days in Southern California, so get out and do that. Head into work. I work on the West Coast at a large pharmaceutical company, and we do early stage discovery toxicology, and trying to find safe and effective medications for people that are sick. So that comprises the bulk of the 8 or however many hour day it happens to turn into, and then it's home in the evening to unwind and just either read a book or pick up a magazine, interact with the family.
I often start off with a early morning teleconference, and then the rest of the day, at least as a consultant, is mostly analyzing data, writing reports.
Every day is different. That's what makes this job so awesome, because I get to see, every day I get to see new drugs being created. It's almost like a cradle to grave thing. You're there in the discovery phase, and you get to see those ideas come to fruition. They come to chemicals. We test them for safety. They look good. We take them into the clinic. They look good. You get them out the other side. They're a drug on the market, and you can say, we've been there from the time that the drug was conceived until the drug, actually eventually goes off the market because something else has replaced it that's better. So, and every day is different. Nothing is the same, and that's why this job is so amazing, because nothing is, you never get bored.
I think the state of the industry that I'm in, the pharmaceutical business, is where I think we're at a point where there is a misperception, a misunderstanding in the public of what we actually do as an industry, and I think that the misperception leads to mistrust of what we do, misunderstanding of why drug development costs are as high as they are. But we are good people. We do good work. We work hard to make sure we make safe and effective medicines for people that are sick. But I think there's a definite gap between what we're able to portray as an industry, how we're perceived by the media, unfortunately, which is where a lot of people get their information, and that's an incomplete and biased story. And so I think that we need to do a better job of educating people on what we do as an industry and how we actually help society and people live better, healthier lives.
We have novel drugs, that came to the markets that have shown that the industry is still very, very innovative. You know, that we do make progress in the industry on diseases that could not be treated so far.
I would say, I mean, it is always in flux.
But so I say that we're in a state of flux, but I look back sort of trying to be in the shoes of, you know, people who were around 20 years ago. They probably would have said the same thing, because they were also dealing with sort of trying to come off the models that traditional drug models to new models, new regulations, new, you know, payer challenges, new, you know, regulatory and political challenges. So, I mean, I think it's probably, you know, we're doing the same thing. It's the challenges are different because we're at a different period of history. But I would say we're in transition like we always are.
I think the industry is doing pretty well. I mean, from where I see it, I mean, all the consultants I know everybody's staying pretty busy, which is a good sign, that there's enough work for everybody. There's a lot of venture capital right now. You're seeing a lot of startup companies. They're very busy. They have a lot of money to spend, or at least so it seems. All the contract labs are very busy. To get a study in, sometimes you're going to have to wait because they're booked up. So to me, it seems like the state of the industry is looking pretty good right now. I mean, I'm not going to say it's as best as it can be, but I think it certainly is doing pretty well.
I think probably the biggest Gordian knot, if you will, is getting enough trust in, or having the regulators trust us enough to know that we're doing the diligence to know that we go into people safely. I think the hurdle around thinking through how we interact with regulators is probably, at least in my mind, one of the biggest challenges.
Convention. Believing that the only thing you can do is what's already in the literature. I think it's good to rely on the literature. It's something that you can learn from. You know, it's like learning from history, really, because as soon as it's published, it's out there, so it's essentially history. But I think what holds us back is believing that that's all that we can do.
I think it just, you know, with many disciplines, you kind of get stuck in a rut sometimes, or doing the same old, or certain departments not talking to other departments to do a paradigm shift.
You know, one of the things that I think about that I wouldn't say it necessarily comes up a lot because not everybody gets there, but would be the 2-year carcinogenicity bioassay and the need for that in filing for registration. I think there's been a lot of data and studies of those that have been done that really show that the data that we get does not translate very well to human tumor incidence from correlating to rodents to humans. So, I mean, that's just one very small example of how there's things in our industry that we definitely need to continue thinking about, and are they really relevant? And right now we do them because the regulations tell us we need to do them. The regulators probably understand that they're not that helpful per se, but they're also not going to tell you can't do it either because, you know, they're all risk averse.
I think also for risk assessment, you know, there's different languages and agendas for all these, and getting them together in a room and then speaking the same language, and at least learning what we can learn from each other and having those communications, which has begun.
I think the absolute biggest one for pharmaceutical science just as a whole is the whole thing about risk assessment.
Really, that history is holding us back a bit. I'd say I said I was in discovery research. On the pharmacology side, people have been realizing for about a decade the role of human genetics in better selecting targets and also in targeted subpopulations for the therapies. We're only now coming to accept that in toxicology, and I'd say there's quite a large component who are still resistant and who feel that the traditional studies that we've run and process that we have is good enough. And I'd say that's a big challenge.
I would say we're limited by the models that we have right now, and none of them, including, you know, in vivo rodent and non-rodent models, can accurately predict for human toxicology. So it's well recognized. It's the best we have right now, and I hope that you have something better in the future.
I think the only limitation is let's move forward, but let's be careful. We can generate, again, a lot of data, but let's be careful about what kind of data we're generating to make sure that we can actually interpret it, otherwise it's a waste of money.
Probably the translational aspect of it. How does what we're doing non-clinically, from a tox point of view, actually translate clinically? Because if what we're doing only translates 50% of the time, you got to wonder about what we're doing. And I don't think we really understand that fully yet. I think we understand it on the backside. If you saw something clinically, did you see it preclinically? Yes, but the question is prospectively, if you see it preclinically, what's the relative certainty that you're going to see that?
The most prevalent that we are aware of, and which is becoming more and more an issue, are the neurodegenerative diseases, for example. So these, and to find treatments for these diseases, I think that's the biggest need, yeah.
So those are the two big barriers. Getting the field to really accept that it's something that's needed, but then also getting the regulatory agencies to accept that and be more open to new processes and substitutions, not just for large molecules, but also for small molecules. I think accepting that, particularly for large molecules, that we don't need to do in vivo toxicology studies would be a very bold solution. And I would say that some companies actually do this. They simply design their drugs to have no species cross-reactivity besides human. And because there's resistance using, say, chimps or greater apes, they never test it in preclinical species. It's all in vitro and target biology based. And it's been okay.
It's not just one, yeah, because there is a lot of, that's disappointing, huh? Sorry for that.
It does take a paradigm shift, and I think of even past analogies, like when I think of how, even speaking of Alexander and other great war generals, that it usually takes somebody to sort of think outside the box and not just keep doing the same thing, because that's the way we always did it.
One of the things that I think is a good thing that we are doing and moving towards is, basically predictive toxicology. I really like that industry, and that's something I'm involved in, the in silico predictions. I feel like the better we can make these computational programs to predict things, the less we have to use animals. Granted, it will take a long time to get there for a lot of these, to where we really, how truly we believe that data that's coming out. But I think, you have to start somewhere, and so we're starting that with genotoxicity. We're doing all the in silico predictions now, and there's some pretty good concordance with that data if you combine that with an actual expert review, and I think that's great. I think it's a great step. Clearly, we need to do a lot more of that, and there's a long way to go, and it's likely not going to happen in my lifetime for any other of the endpoints, but I think it's a great start.
I think the challenge would be to, maybe if there is one thing, then to make this more streamlined, if you like, that these steps are like nicer connected with each other, and in the sense of nicer means more relevant to each other, yeah.
If I could change anything, it would be that shorter-term studies were more predictive of longer-term outcomes, so that you reduce the amount of testing, animal testing, that you have to do.
But we have to be willing to accept some level of risk to reap the rewards, just like they do for treatment in certain indications, which are underserved.
A bold solution, fully integrated organ models, so that we could study essentially whole body toxicology on a micro scale. That's a bold solution. One-stop shop.
I think that goes against the nature of a toxicologist, which is to guard those volunteers' health to the best level we possibly can, and it just feels super uncomfortable. But we're going to have to do that if we want to really make an advance and gain experience with it.
I guess my biggest aspiration would be to have toxicology not be held in the strict old standards, for it to be allowed to break out of the box.
I think it was a mathematician who once said, all models are wrong, some are useful. So, you know, what that means to me is that anytime we're looking at something that's not the actual system you're looking at, you know it's not going to directly mimic that system. And so, keeping that in mind, if you ask the right questions carefully, you can get answers that might help you make decisions in that ultimate system. So I guess I would say it is probably another model that is going to be wrong, but it could be useful if it's done thoughtfully.
That the in vitro and pharmacogenomics, toxicogenomics, proteomics, alleliomics would be suddenly found to be, we have gene signatures that are in fact predictive of a tox outcome or an efficacy outcome, and that you could actually just take a lot of your tox that maybe come from your efficacy models, and you could say, when I see this, I know it's going to work, and it's not going to have that particular toxicity, that you could actually tailor medications more towards individual populations of people.
And I think we have tremendous abilities to generate a lot of information, but it needs to be interpretable, it needs to be useful. We need to be able to find the nuggets in there that make sense and will be really informative for the endpoints that we're interested in.
There's this whole initiative for the 3 Rs right now, and reduce, refine, reuse. I think a lot of that has to do with the use of animals and finding alternatives to subjecting animals if indeed you don't have to, because you have relevant in vitro models that can do the same type of assessment for you. And I think that's where the field is going. That's the motivation. When I go to meetings, that's what I'm always hearing about, is application of the 3 Rs for toxicology.
Put yourself in a situation, pretend, if you will, with a project team. We have an urgent medical need. In fact, it's so urgent, let's think of it like an outbreak of a serious virus. And we need to get something done this week. What could we possibly do in a situation like that? And then just have some group think around, put it in the situation where you're desperate, and then let's have some ideas around that.
Whether they're your basic, bench level toxicologist generating the data, or even an in vivo toxicologist that's being told they can't do animal studies anymore. So in a way, we've been told you've got to change the Gordian knot, or you got to untie it no matter what.
In some cases, you're just not adding value. You really just need to be bold.
The short answer to that is no, because you kind of have to understand the mindset of each of those and what, I guess, for lack of a better word, what they trust or have faith in. And so if you're a traditional toxicologist that kind of grew up in the traditional in vivo type, especially animal model type of toxicology, then you don't trust anything that an in vitro model can do.
I think at least in the organization that I'm in, we're pretty closely integrated. I think if there's any sort of division or silo, it's between non-regulated type of studies and regulated studies. And so that's one distinction that exists and probably will exist for a long time. Probably the key area I see for mutual discussions on the topic is when our in vitro models just don't recapitulate what we see in vivo. I think that's where there's some fundamental lack of understanding of mechanism, lack of understanding of the complete biology, where the simplified system is just a little too simple.
I think they do have a divide, and you see that it's not just toxicology, you see that in the world of drug discovery as well. It's a chasm for multiple reasons. A lot of it has to do with the fact that a lot of in vitro folks find in vivo work uncomfortable. They deliberately choose that they will never touch an animal and only work with cells. And so right there, it's sort of an affront to in vivo folks, a little bit of discomfort, because you never know when you're approaching someone with an in vitro background if they're uncomfortable with you simply because you handle or work with animals.
I wouldn't say there is a divide, right? These people have to work together, you know. So, but you're right. I mean, and certainly how we do it today, I don't think it's the best way, obviously. And ultimately, in toxicology, we should not be using animals, right? Because, again, like I said before, and many times before, is like, we want to treat patients, not animals, right? Not rats.
I think there used to be a gap, but I think the gap is narrowing because I think, because of this translational aspect of it, so I do think that the gap is beginning to narrow, and I think there is more as we see where some of the in vitro stuff can potentially become translational. I think that will actually increase over time.
But there's a deeper one in toxicology, and it has to do with, you know, this question about whether individual assays, whether they're biochemical or cell-based, can truly represent the physiology of an animal, and that the only way to really know is to dose the animal. And okay, there's certainly you know, points to that, but you can also often predict what's going to happen in that animal through those in vitro measures. So I think combining them, you have to. Certainly the detective work that gets done from an in vivo study is all in vitro, or at least biomarker based, which relies on in vitro resources. So you need them to work together to deliver the best package at the end and come up with the best plan.
I think it all comes down to model validation and being able to demonstrate to a scientist that whatever the model is that you're using replicates what's going on in a human system. I think, to me, that's the ultimate goal, or should be the ultimate goal of any in vitro system. The closer you can get, and part of that is using just human cells versus animal cells. To me, human cells have more value than animal cells for assessing safety. But beyond that is, how do those cells in the dish react versus how does a whole organ react? And how can you validate and show that what you're doing in the dish replicates what's going on in an actual human?
I think we need to come up with ways similar to what your organization's working on to just kind of dial that out and go, let's, even though we need simplified systems, let's do it in a manner that pulls in all the knowledge that we currently have.
You have to combine the group, and you can't keep it separate where there's a in vivo person who's in charge of the in vivo folks and an in vitro person in charge of the in vitro. It doesn't work like that. You need to have them intermixed with one another.
So to me, the more predictive toxicology that we can do would be the best thing, in my opinion, to kind of move it along. I mean, granted, there's many other things that we can do and work on, but that's just one, and that's kind of my particular interest.
I really would say I'd like to see toxicology have a stronger seat at the table in terms of what goes into the clinic, and not being so willing for folks to downplay the very real potential of toxicity to halt a program in the clinic, not just lack of efficacy.
To be a toxicologist, you need to know chemistry, you need to know biology, physiology, because it all kind of meshes when you're trying to assess the safety of a drug. So if you get bored doing one thing, then you might want to consider toxicology, because there's a lot of avenues that you can take.
I would say that it's definitely a good career path, a lot of promise, and not something that the mainstream is thinking of all the time, which sometimes is a good area to focus your efforts. Yeah.
Toxicology is cool because it's not just biology, it's not just chemistry or biochemistry or physiology. you have to know a little bit about all that stuff to be able to bring it together, which is why I think toxicology is really cool.
For me, it's been an exciting and fantastic journey, and the best part about it is I get to do something new every day. Even if it's in the regulated space where you are working in GLP studies, you never know what that next day is going to bring on a study. So that's, again, part of it that's just, it's exciting. You know, it's a discipline that we get to interact with a lot of other colleagues within the company, you know, colleagues in early drug discovery all the way through to people that are thinking about how to market a molecule. And the data that you generate in a toxicology study impacts what that label looks like. And so things like that are just, I mean, to me that's really exciting, and why would you not want to do that every day?
Because I think right now the field of toxicology is wide open.
So get ready, it's a wild ride. You never know what's going to come. You're going to have to talk to some people and manage their expectations and manage their assumptions in a way that can be very challenging at times about what is the role of toxicology in the drug development process. But you're also going to learn a lot and have some great colleagues and some fun along the way.
You know, you can start really young with having a great interest in science and in toxicology, but you really need someone to open that door for you and let you see the possibilities. So I think my advice really is to the seasoned scientists out there, because there are a lot of people that have an interest. They just need to know what the possibilities are. So I think it's more our responsibility to the next generation to kind of let them know what those possibilities are.
Oh, I would tell them it's exciting. It is something different every day. I love going to work. I've never had a time where I thought, oh my God, I'm bored. You can do just about anything you want to do. If you really want to focus in the lab, you can focus in the lab. If you really don't want to do that, because some people don't really enjoy that, you can be solely focused on regulatory review. There's so many things you can do, and what you're doing actually is bringing drugs to market for people. So you're actually helping, you know, people. If I were a tree, what kind of tree would I be and why? I would be a chimera. I would be a combination of an oak tree and a maple, a sugar maple. And I think that I would be that because oak trees are strong, they're long-lived, their roots run deep. But I would want the maple because it's colorful, it changes, and it really probably reflects my personality, you know, and it's more reflective of who I am.
I would be a birch, because the birch are first of all, I think they are beautiful trees, right? I mean, the stem, you know, with the white and the patches, I think that they are very nice trees, you know. They are also the leaf trees, you know, so that means they change their colors, you know, they get renewed, you know, like.
I would be a magnolia, because I am from the South, and so a magnolia represents to me being a Southern belle.
Well, happens to be, I am a tree, so this is easy. No. What would I be? I don't know. I guess I think I'd be a pine tree, because, you know, it's always green and it smells great. So that sounds like a good good thing to strive for.
It's actually very easy for me, that particular question. I would be a willow tree because they bend and flex with the changing environment, and that's how I see myself. I'm also very balanced, but I practice martial arts, and our theme is the willow tree, and so I've done a lot kind of particular to that theme. So for me, I would be a willow tree.
I would be something tall overlooking the coast that sometimes gets snowed on.
I would be a willow tree. I love willow trees. I grew up in the South, where we have quite a few, and there are many poems written about the elegance and beauty of willows. But having spent time under different types of trees, I can say the willow is always my favorite. You have shade in the summer, you have beautiful music and wind and storms, and in the fall you really don't have a lot of leaves to clean up. It's actually a low leaf tree for all that it's shade, and you don't have tons of pollen blowing on you like dogwoods or cottonwoods or other trees that make it look like it's snow in July. And so I just find it to be a beautiful and welcoming tree.